Like, that's my home. No, I feel like
Yes.
that today,
Yeah.
the stuff that we were learning last week.
Yeah, it's right there.
It's like, yeah, yep, there's a wonderful thing. It's never been happened. But then, oh. One minute. Yeah. Oh, no, that would be exciting. Then I got. I hate that. You're like, I You do so good. Until 11:38. Yeah, like, so it's like, no, I think it's. All the. So they look at.
Okay, lecture 9. Let's get started. So there should be everybody get a handout who wants one. There's extras up as people come in. Make sure they point out, pick them up. First exam, 2 weeks from today. Looking at your equine science book, they do a pretty good job with bones in chapter 3. But we'll focus on what we talk about in class. So last time we were going to start composition. So we're looking at bone. It's an active living tissue. What is it made of? So we're going to be really simple here. We've got matrix and we've got cell. Matrix is the bone, the protein, the calcium that we think about in bone, but we also have the cells that are doing all the work. Remodeling, fixing, building up, tearing down. If we're looking at the matrix, we have the organic components. So what do we say about organic components? Anybody remember how we define organic? Yes, sir. Carbon has carbon in it versus inorganic. The two primary ones we're dealing with are calcium and phosphorus. Where did we get those from? What group? Macro minerals. So the big four: macro minerals, micro minerals. There are a bunch of other minerals there, but we're going to focus on calcium and phosphorus. The organic, we're dealing with mostly proteins. We're going to bring in some sugars and stuff to help with the ends of the bones, to help lubricate things. But we're looking at a protein matrix. So last time I mentioned that if you take a chicken bone, you soak it in vinegar, in theory, everything gets floppy because you take out all of the inorganic components. That acid in there puts the calcium in solution and then leaves you with just protein, and you got something that's floppy. Then we've got 4 cell types we'll talk about in this class, and we'll explain what each of those are as we move forward. So osteoprogenitor, osteocyte, osteoblast, and osteoclast. Each has a specific function, a specific purpose inside the bone tissue. So bones, if we're looking at most of the weight of a bone, we're looking at the inorganic component, the calcium and the phosphorus. That's going to account for most of the weight of our bones. We're going to have most of that is in the calcium hydroxyapatite form. We have a ratio in of calcium to phosphorus about 3:2. Anybody here in nutrition and you balance rations usually have about twice as much calcium in the diet as you do phosphorus. That comes from this relationship right here. So calcium, 10 of those. The phosphate groups, you're going to have 6 of those. And we've got a couple of hydroxides attached as well. We can also replace those hydroxyl ions on the end with either a fluoride, a chloride, or a carbonate. So the hydroxyapatite is going to be the dominant mineral that you're going to find in bones, followed by the replacement, the calcium carbonate, replacing one of these with a carbonate CO3 molecule. That's going to be somewhere in that 10 to 20%, depending on where we are in the life cycle. Most of the volume of the bone is going to be matrix. We have that protein skeleton that determines the shape, and then the body adds the inorganic components to make that shape rigid. Has anybody heard of rickets? Your animals don't get enough calcium or enough vitamin D, they get rickets. Does anybody know the characteristic sign for rickets? Legs tend to fall out. So if I don't have enough calcium or I don't have enough vitamin D to calcify the tissues get floppy and they bend. They bend out. So we want those bones to be well calcified and rigid. The exact mixture or relationship between those two is very dependent on age. I think of kids running around all the time, bouncing off the ground, get right back up. If I did that, I would break something. They have more matrix. They're more flexible in their bones than I am. I'm pretty well set, and I've got as little matrix as I'm going to have. Of course, as time goes by, I'll probably start losing that matrix and the calcium as well. So if we look at a cartoon of the relationship, we have a long bone cartoon here. That's in utero, an embryo, fetus. We have a young person here, and then we have an adult over here. If we take the blue as cartilage, we have something that's not yet ossified. We haven't put the inorganic components in and made that rigid. And we'll talk about how these bones grow in a minute, but when we're younger, we have growth plates, we have ends that are expanding. So we start in the middle here, we grow out from the center of the bone. We have a growth plate here and a growth plate here that's going to continue to build up and make that bone longer. Once we go through puberty, the growth plate, which is right here, ossifies. You're as tall as you're going to get. Legs aren't going to get any longer. So it ossifies. We bring in that inorganic, the calcium phosphorus component, and the cartilage we have is at the end in the joints. So over time, you're going to get more and more inorganic components replacing or ossifying those protein components. So again, as we get older, we get more and more of this added in to the structure we've established already. So bone types, we mentioned the 4. So we'll start with the osteoprogenitor. There's a couple other names you'll see in the books and some of the diagrams coming up, but it's a bone stem cell. Does everybody know what that means? Does everybody know what a stem cell is? All that controversy in the news and stuff, whether we should use them, whether we shouldn't use them. You got a cell that's still in a generic form that hasn't completely differentiated. With our osteoprogenitor, we've got something that's differentiated from the generic cell enough to say you are going to be a bone cell. Which type is yet to be determined. So we have them in there, and they can turn into any of the next 3 as we go through. So we'll start with the osteoblast. The osteoblast builds bone. It'd be really nice if they broke down bone. You could remember that really easy. Osteoblast, B for build bone. This is key in remodeling. Its pair in that activity is the osteoclast. Got osteoblasts, they're going to build bone. Your osteoclasts, I use a C to clear bone. So if we're going or exercising our horse and that horse is going to build up bone to resist breaking the stresses that it's going through. Both of those yin and yang are working together to make the bone as light as you can make it and as strong as you can make it. The osteocyte is key in calcium balance in the body. We think about the bones as a reservoir for calcium, phosphorus. When we need those things, we take a withdrawal from the bank. Calcium, the osteocyte's there to remove that calcium when the animal needs it. If we have extra, particularly in the summertime, we're going to put that in the bank for later. So you can think about the osteocyte as the bank teller or the ATM. I can make deposits, I can make withdrawals. Any questions so far? So this is another diagram here. You don't have this in your handout. Again, osteogenic fibroblasts, they can become any of the other three. So blasts are going to build the bone. Blasts are going to take it apart. And then we have the osteocytes that sit in our bone and add and subtract as we need to. If we start to look at the whole, there are two different types of bone tissue that we need to be concerned about. One is compact, one is spongy. So compact will also be called hard, dense, or cortical. What does cortical mean? Cortex. Outside. Then the spongy bone, cancellous or trabecular. So compact bone is usually on the outside of our bones. We have a denser shell, and the spongy bone is inside of our bone. A matrix there to kind of provide some internal scaffolding, give some bone some extra strength with a lightweight approach. So if we're looking at a generic long bone, this is suggesting it's an arm bone. So we've got the compact bone around the outside, and then we've got our spongy bone in the center. So you've got more or less continuous bone matrix, organic on the outside, and then on the inside, you've got a scaffolding that's going to help support that outside, help that bone resist strength as you go through. So we call it spongy bone. Why do you think we do that? You think it's soft and squishy? It looks spongy, but it's not. It's a scaffolding. It's a, rafters that kind of hold everything together, but it's full of holes, and that's where its name sort of comes from. So that's a stereograph. If you can get one image in one eye, one image in the other, you can see the 3D structure that's cartooned there. So similar to a sponge. The other we're looking at is the compact bone, and this is sort of a microscopic view of that. I'm going to look down on the wall. So we got maybe the outside is up here and the inside is up here. What we're establishing, if we don't worry about names for a moment, you've got these cylinders of bone matrix, and they run kind of the length of that compact bone, that bone on the outside. In the center, we're hollow. That's where the blood vessels go. We need to be able to supply the tissue with nutrients, be able to supply it with oxygen, take back all the waste, carbon dioxide. All the little black dots here are osteocytes. So what do you look at from a complicated or what do you see that automatically complicates the life of an osteocyte? Most of you, if you were taught anything, cells are right next to capillaries, right? So they can get supplied and waste can be removed. We're starting to separate things from their blood supply. If we look at a cartoon of that same thing, we were looking down on the top here. We've got our cylinder, and then these little spider things are where our osteocytes are sitting. They're going to put calcium in when we have extra. They're going to take calcium out when we need them. So you have the blood vessels running down the center of these osteons. Os meaning bone. What's the problem or challenge for our osteocytes? They're way away from the blood, relatively speaking, in the body. Every one, you can see kind of up top here, there's a canal or a hole that connects them. So you have an osteocyte, and you can yell at the neighbor through a hole, "How you doing?" through that hole. So all the nutrients have to move through all those little holes, those little canals. You can see them vertically here. So things have to diffuse out from the blood, work up and down through these little canals to provide nutrition, take away the waste products for these cells. That's a little harder than most of the rest of the body. Things are a little bit, time is going to take, it's going to take more time to get things in, get things out. And this is one of the reasons that bone heals relatively slow. You have to establish all this structure, the matrix amount of living cells, and you have to diffuse everything in and diffuse everything out. So processes there tend to be a little slower. So again, another cartoon of that. We're looking down on the osteon. We've got the canal here in the middle that has our blood vessels. They're usually paired. Veins and arteries. The little red things here are the osteocytes. And then all the little purple pathways are the canals that connect them to the blood source. So you got a little small hole. Everything's got to come in through those pathways, and it's got to go back out. Another drawing of those. As we get here, you see the establishment of the osteocyte, the extensions they have to deposit and withdraw calcium. We're just getting started here. We're establishing here. You'll also see as we remodel that one area might start to rip apart one area and rebuild, so we get these overlapping structures. Generally, we start from the inside, we take everything away, we put it on the outside. That's how our bones get bigger in diameter. We take things from the middle, put it on the end. That's how we get longer. So creating these things, you're going to have the canal systems, the osteons are going to start to overlap. Some of the newer ones are going to destroy the older ones as we remodel. So again, this shows the blue and the red were paired throughout. So you have veins coming in, arteries going out. So you've got a lot of blood through this system. We're on the outside here. And then we got a spongy bone on the inside. We've got blood vessels coming in. We get to our osteocytes sitting in a lacuna. And we establish those systems. Another drawing. I don't think you have this one in your handout, but I liked it, so I added it in. So you have that drawing. We see from the outside all the blood vessels that supply a bone come down the outside and work their way in. We don't want them coming through the ends, right? Because if the ends are moving, we're going to shear those blood vessels off. So they're going to go parallel and then come in. Each osteon has its own blood supply, and we service a number of osteocytes in that osteon. So we gather those together. That becomes our compact bone on the outside. As we grow, we eliminate the osteons on the inside and we put more on the outside. So we get a better, bigger diameter. And as we build on the end, we'll get there. So another view of the same stuff from the 8th edition of Anatomy and Physiology of Farm Animals. So bone's going to serve a number of diverse purposes. For a moment, one of the basic rules in A&P that almost everything in the body has 2 or more purposes, especially when we get down to the elements. Calcium has a number of different functions. Phosphorus has a number of different functions. You get down to your trace minerals, sometimes it's only one thing. But sodium, potassium, calcium, phosphorus, sulfur, magnesium, and chloride all have multiple functions. So with calcium and bone, we're thinking about providing structure, protection, and support for the body. We're housing the bone marrow in that spongy bone. That's where our blood cell manufacturing system is. We're going to store for later use or times of shortage calcium and phosphorus. And as we mentioned before, we have that yellow fat in the bone marrow that will serve as an energy reserve, particularly as we manufacture those red blood cells. So pulling out a little bit, you've got the long bone here, the marrow inside. We'll have a variety of cells, red blood cells, erythrocytes, the white blood cells, the leukocytes, and the platelets are all manufactured inside that bone. So if we're looking at a long bone, we need to get some vocabulary down. We have an epiphysis at either end of the long bone. The one closest to the body would be called the what? Proximal epiphysis. The one furthest from the body is the distal epiphysis. So any of our long bones has an epiphysis on either end. If we're looking at a generic bone here, you've got one proximal here. It goes down to our epiphyseal cartilage, or epiphyseal plate, growth disc, and then we got the distal, again, separated from the rest of the bone by that growth plate. The diaphysis, the center or middle of the bone, so the center section. So we're looking at probably something from here down to here. The last part we're going to talk about is where the flare starts. So the metaphysis is between the diaphysis and the epiphysis, and it's the flared section in that long bone. So epiphysis up here, we start to flare out. We got our metaphysis, diaphysis in the middle where things are pretty much uniform in diameter. We got our flare down here at the other end, and the distal epiphysis at the other end. Another diagram that goes into a little bit more detail. I provide it for you in the handouts when I post it, but we're not going to get into detail right now. Separating the epiphysis and the metaphysis is the epiphyseal cartilage, disc, or plate. This is the area where the bone grows from when we're thinking about the extension of it. So it's a layer of cartilage that separates the cap or the epiphysis from the shaft of the bone. It's found in immature bone, and it's the only spot where you can extend the length of the bone. So that growth plate here, actively growing, making that bone longer. As we get to adult, we get through puberty, that growth plate ossifies and our upward growth is stopped. All I've got left is I can grow out. Can't grow up anymore. Another look at this. We're sort of maintaining three separate spongy areas separated by the bones. Each has its own blood supply. So again, those vessels run along the bone and then come in where they need to supply them the diaphysis or either epiphysis. So has anybody here heard of Osgood-Schlatter's disease? Has anybody here broken the top of a long bone as they were growing? But one of the problems with that is if you break that top of your long bone or either end going through the epiphyseal plate, you're going to prematurely ossify. What's that going to lead to? One leg's going to continue to grow, the other one's not. I had a friend of mine's father that broke when he was a very young man, before the science they have now where they can keep that plate growing. So you keep your legs relatively the same length. He had a 2-inch difference between his legs because of a fracture he had in his single digits. So if that plate breaks, we're going to prematurely ossify it, and we're going to stop growth on that end of the bone. So we don't want to break this before we're done growing, or we're going to get some unevenness in our long bones on either side of the body. So that growth plate, when we're talking about growth, we're talking about bone formation, we start with a plate, and bone tends to grow or lay down here, and then here, and then here, and here. We kind of build up to get that long bone. The pattern we're dealing with is first we establish the bone cells in that area. Then we establish a matrix. Then we ossify or make that matrix rigid. Another view of the same sort of thing. We're getting our bone cells lined up. We start doing, does everybody know what mitosis is? So dividing and multiplying a cell, this proliferative zone, we get more and more cells there. We divide, then we hypertrophy. Does everybody know what that means? Hypertrophy means we get bigger. So each cell gets bigger and bigger. And during that phase, we're laying down the protein matrix, the shape of the bone that we're going to have. And then once we get that done, the ossification zone occurs. So you can see that over time, these zones are going to move up, and this calcification zone, as this is completely grown, this grows up, that ossification zone is going to build up, and we're going to move toward the growth plate up there. So you're in here, you got your long bone here. We're going to use that pattern over again. So this is a micrograph of that. We got the zone of proliferation where we're dividing our cells. We start with a osteoprogenitor type cell up here, and we keep dividing. We start to specialize down here, we get bigger and bigger. Their job is to secrete that protein matrix. And then we ossify that. And as we ossify it, we start to lose some of the cell. Digressing for a moment, your epiphyseal plate closes when you go through puberty. You'll get a spike of estrogen or testosterone at some point at puberty, and you're as tall as you're going to be. That plate will ossify, and once it's closed, the long bone growth is complete. So what does that mean for those animals that don't have their gonads intact? These long bones will continue to grow, and then you get something that looks like this. An ox, we take that, castrate that animal fairly early, we're going to get a lot of long bone growth, and they're going to be a little bit taller than if we left that male intact. I know that happens with our ox, but I'm told it also happens the same with geldings. They've done some research there that they're saying a half-inch difference when they're doing a paired sibling study. So siblings that are castrated tend to be a little bit taller than the stallions that remain intact. So we have those on, we don't get that ossification or closure of the growth plate as soon as we would with an intact male. So we're going to have a little bit more long bone growth, a little bit taller. If we're looking at plate closures, so humerus, 3 years, we start to get in those 2-year range when you're really starting to go through puberty. Some of the other ones will stretch out a little bit longer depending on how they react, but that cartilage will ossify and we stop doing growth. The bone is very active tissue, so constantly being formed and resorbed in or reshaping itself with the goal of basically use it or move it. If it doesn't serve a purpose, we're going to clear that bone and we're going to put it somewhere else. So everybody knows about the 2 astronauts that got stranded up at the space station. They weren't planning on being there. What, they've been here 8 or 9 months now. They expected to be up there a week. They're only up there, they're up there for 9 months. Because they don't have the effects of gravity, and this is a concern for long-term space travel. If we don't have gravity constantly pushing on our bones, we're constantly resisting, trying to stand up against gravity, we tend to lose bone mass. It tends to become less strong over time. And I would imagine when those 2 astronauts get home, they're going to have to behave themselves for a little while because they weren't planning on being up there. So I'm sure they do some activities up there. So we use it or move it. The goal is maximum strength with minimum weight. If I made my bones solid, they'd probably be a lot stronger. But then I lose some of that multiple functionality. They're way stronger than they need to be, and now I don't have any place to manufacture blood cells. I have to go somewhere else. So this is how it all gets done. So if you're really interested, you can zoom in on that. That's way more than we need to know in this class. So let's talk about our osteoclasts. They're going to clear bone. Osteoblasts are going to build bone. They work in concert. Clasts are going to take where it's not needed or where it's broken. And then our blasts are going to build that up in another location, trying to reinforce micro cracks. So one of the reasons we start out slow with exercise for us, for our horses, is usually when we start, we're putting a lot of stress on the bones. We get little micro cracks. We do overdo it, we can lead to a big break. But if we build up to it over a few weeks, the osteoclast and the osteoblast can respond and build that bone up the way it needs to be for whatever activity you're doing. So figuring out, okay, we're having some problems over here. We need to build up this area better. We're not really using this area over here. We can take it away and move that calcium over to another place. So we're looking at normal bone remodeling. We're going to have our osteoclast taking an area away. Then we come back and we reform with our osteoblast, reinforce, make it stronger, make it better than we had before. So as we get cracks in our osteons, there's an opportunity there for our bones to be repaired. So when we got that remodeling, the stress, weak areas, areas with micro cracks or areas that appear to be weak are reinforced. Unused areas are dismantled. So you're going to take out the inorganic matrix. You're going to take out the protein matrix, and you're going to recycle them into other locations. Break it down, protein back to amino acids. Going to transfer them over to areas where they're needed. So the ability to do these changes with maturity. So I don't have the ability to remodel like you guys do. My bones are kind of set. They're kind of in the way they're going to be. As we go to maturity, this remodeling can slow down. So remodeling can shrink bone, get rid of it. It can make it bigger, hypertrophy, increase in size, repair breaks, and rearrange the internal structure of the spongy bone to resist stress. So if we're looking at an animal, the effects of exercise, we've got a confined animal here and an exercised animal here. We don't need that bone to stand up in a stall and do nothing. But if we're moving around, we got percussive exercise, we need a thicker outside compact bone. Depending on which way the forces go, we're going to maybe on the front put a little bit more or maybe less. So a normal horse here with race work, this particular bone is going to put more on front to resist whatever problems we have or the forces that we're dealing with. Whereas in the back, we might be something similar to what we started out with. So working our way up, not doing it all at once, training that animal is going to take us from here to here. And we're going to focus on our osteoblasts, the ones that build bone, to have them create something that should last and hopefully resist the forces that are in place. So the bone's going to react to the stresses that are put on it. So racing is probably going to be different than jumping. It's going to be different than our draft horses. Depending on what the strains are, depending on what the stresses are, the bone is going to react differently and remodel to, again, make maximum strength with minimum weight. So thinking about breaks in bones, big ones, little ones, we have a pattern of behavior here that's very similar to what happens at the growth plate. If we have some sort of break, we have our stem cells, our osteoprogenitor cells coming in. They're going to start to put together a soft matrix that protein, the cartilage listed here, to reestablish what the scaffolding should be. What is the shape of the bone? And since it broke a little bit, maybe we need to do a little bit more on both sides to make that bone a little bit stronger. Once we get that scaffolding in place, that matrix, the organic matrix in place, we have to come back with the calcium and phosphorus to ossify that area. So the osteoblast, putting in the mineral, making sure that bone is strong and mended. Then the bone fills the break, and hopefully we have something stronger and stiffer over time. So if we're thinking about breaks, no matter how big they are, what do we know when they first start to heal? What do we know about protein? Or conversely, why do we put your leg in a cast when you break your leg? So it doesn't heal wrong. It's an opportunity here. This is a slow process because of getting nutrition through those osteons, getting the blood vessels reestablished, getting the diffusion down all those little canals takes time. Also, if we have all cartilage here, protein here, it's soft. It's going to bend. We need to have things immobile until we get over here where we're adding that calcium in so things become rigid again. If this bone keeps moving, or both bones next to each other, that scaffolding is going to continue to break, and you're not going to be able to establish the right kind of shape, the right kind of formation in that bone so it heals properly and effectively. So we need to take that time and give it that rest so we're not putting too much stress on a structure, the protein or the cartilage, that can't handle it. So it's the same pattern as growth. We've got the protein matrix comes in first. We've got the ossification, the minerals later, and then we finalize our structure, and hopefully everything goes together the way we hope it will. And there'll be a little extra on the outside because we overdid it. That's an attempt to reconcile that force. So again, another set of diagrams here. You've got your soft connection, you're building stuff around, and then we start to ossify that until we get the hard, completely healed bone. And then as time goes by, depending on what the needs are, the osteoblasts and the osteoclasts will tend to remodel. So looking at calcium's other functions. Again, we said we're going to use the bones as a bank. We're going to use calcium to help manage our blood pH, help manage our blood osmolality. It's going to be a secondary messenger inside the cell. So it's going to be linked with hormone cascades, nerve impulses, and muscle contraction. We need calcium for muscle contraction. It's also involved in blood clotting and immune responses. So if we're looking at calcium homeostasis, one of the challenges with calcium is it's very insoluble in water. Does anybody here get orange juice with extra calcium in it for your bone? Anybody get it? Well, that's a good thing, because if you get orange juice is a little acidic, and if you take that, let that sit in the refrigerator for a while, take it out, slowly pour it out, all the calcium's on the bottom. Now, when they test it, they have to shake it and do the standard lab stuff to get it in solution for a couple minutes. And then they test, well, that's how much calcium, but it's all in the bottom because it's very insoluble. Orange juice is slightly acidic. We also find that calcium is pretty scarce. When we looked at what makes up the Earth's crust, calcium is not one of the top things. So when we get it, we need to store it for later use. We need to have it around when we need it. So the bone acts as a reservoir for calcium. So reservoir, when calcium is needed, our osteocyte's going to pull calcium out of the bank and put it into our blood when we need it. If we have extra floating around, we don't want that precipitating out of solution, the water in our blood, and calcifying everything. We want it to get out of there, and we put it in the bone. So calcium in and out of the bone is controlled by the osteocyte and two hormones. So you've got parathyroid hormone, PTH. That's going to be responsible. Calcitonin. Both of these hormones come from the parathyroid. Does anybody know where the parathyroid is? Got one on each side of your windpipe. So there's the parathyroid senses the calcium levels in your blood and decides which hormone to release depending on what we need. Those two hormones will go to the osteocyte. So what is the challenge of getting those two hormones to the osteocyte? Got to get in the blood, you got to get in the bone, you got to get out of the blood vessel, and then you got to go through all those little canals till you get to an individual osteocyte. So this is not a fast process. It takes a little while with the diffusion through those canals of the osteon moving through. So we'll stop there today. I apologize for not having these on the handout yet.